Hey parts, it's Professor McNeil here. We're learning about meiosis today. I put together a short little video for you just so you could understand with some nice diagrammic pictures that we've got up here. First step, prophase 1. Now, prophase 1 is just you see the spindle being formed here. It'll be shifted in further steps of meiosis. We see the sister chromatids and the homologous chromosomes crossing over, which makes every single one of us different. Look at Wells and look at Dawson. They're two completely different people, and you want to know why? Bam, that's why, crossing over. You see, we get the crossing over takes place when the sister chromatids interlock. And you can see that right here in this diagram. We're going to move on to metaphase 1.
Professor Owens, so you're talking about the second step of meiosis, which is metaphase 1. During metaphase 1, the microtubules become attached to the chromosome, and this point is called the centromer. See, these things get very attached, way more attached than we get to women. What can we say? We're players. Just look at us. But yes, once again, they become attached to the chromosomes, the microtubules do, and very attached. I'm going to reiterate this, way more attached than we get to women. We are players. Keep that one in mind.
It's Professor McNeil again. We got the third step of meiosis, anaphase 1. We're getting into some risky business, if you know what I mean. We'll see here. We got the spindle is formed. We got the sister chromatids remain attached, but they're being pulled apart. You can still see the crossing over in the chromatids here. They're being pulled apart. The homologous chromosomes are being pulled apart. They're separated, being pulled to opposite sides of the cell here. This is basically the warm-up step for the next step we have, the last step of meiosis. What? Yes, question. Owens.
Do the sister chromatids, do they ever come apart? Because I'd be kind of sad to see that because, sisters are really close and all, and you should never want to see them torn apart, man. It gets sad, man.
No, I've got a good answer for you. They will never come apart. They remain attached through the process.
Thank you. Telophase 1, fourth step of meiosis. It's where the cleavage furrow occurs, which is the start of the splitting of the cells. The cleavage furrow is a result of cytokinesis. And no, Luft, I see you on the edge of your seat back there when I talk about cleavage. And it is not the kind of cleavage you would think. Not the one that we know you so desire. Oh, God, Luft. Yeah, it's the one that occurs in animal cells during cytokinesis. Yes, McNeil, question.
Professor Owens, I'm looking at this diagram and I'm seeing this odd shape. And what is there? Are they coming apart? Are they about to separate, or what's happening?
They're about to separate. See,
okay.
You can see them pinching.
Okay.
It's starting to pinch.
Oh.
Yeah, like when you get a grape. Pinch it, comes together.
Yeah.
Splits.
All right, perfect.
And edit.
Hey boys, Professor McNeil, meiosis 2, prophase 2. It's getting serious. What just happened in telophase 1? We had a separation. Now we've got a start of 2 cells here. We've got our homologous chromosomes, our sister chromatids crossed over, spindles form. I can't wait to see what happens in the next step.
Go. Professor Owens here, guys, talking about metaphase 2. I know, one of my most favorite steps. Yeah, it's pretty much the same as metaphase 1, but now it occurs in not 1, but 2 cells. 2.
Nice.
And so it's just very impressive. They once again become attached to the chromosomes. See, the microtubules become attached to the chromosomes here. And we made that knee slapper joke about us being players. Ha ha. Just kidding. It's not a joke. We are players. Okay? Get that one through y'all's heads. I mean, and that's about it with this one. Can't wait to see what happens with the next step.
We're back. Anaphase 2. We got the sister chromatids. They separate here, moving across, kind of like a magnet. Let me put an image in your mind. Magnet, pulling apart, just pulling things away. That's what we're happening here. We got one more step after this. You can see that the oval shape is coming into play, meaning that they're about to separate. We got the sister chromatids moving, and you know, on this one we've got we've got the sister chromatids separated instead of the homologous chromosomes like in metaphase or anaphase 1. I'm sorry. I did lie to you. The sisters do separate. Tough luck. And that's all I have to say about anaphase 2. Can't wait to see what telophase 2 has in store for us. Thank you.
Professor Owens here, and I'm sorry to say, boys, this is the last step. I know y'all desire so much more than this, but it's okay. See, in this step, a cleavage row occurs again, pinching the cell into not 2 diploid cells, but into 4 haploid daughter cells. Whew, God, just gets me nervous talking about it, man. And once again, Tim, get back down in your seat. This isn't the cleavage we're talking about that we know you want. Okay, so just chill out. Okay, and see.
Yeah, Tim.
Tim. God, Tim, just go back. See, they're pinching here, and once again, a cleavage row occurs, which happens in animal cells during cytokinesis. And just to reiterate, it is no longer 2 diploid cells. It is now 4 haploid cells. We went from 1 to 4. Tense. And that's all we got. See you pards later.